Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. So today I've heard is something called Liberation Day. And it's a day when I'm supposed to announce tariffs on people. But instead of announcing tariffs, I think I'm going to just share some very good science with the help of our special guests today, Tamás Visnovitz and Edit Buzás. Um, so we have a new form of EV release that we're going to be discussing today, and thank you all for joining. During the presentation, I'm going to have everybody on mute, but I will ask you to put your comments and questions for Edit and Tamás in the chat box, and then at the end of the presentation, I will allow unmuting and invite you to share your camera if you'd like, and also to speak with our guests. So I'm going to turn this over now to Edit Buzás, whom I'm sure that all of you know very well as the former president of the International Society for EVs. Edit is at the Semmelweis University in Hungary. And Edit, I'd like to invite you now to introduce this project and its origins. Thank you.
Thank you so much, Ken. Thank you for the invitation and for the opportunity to discuss this project that we have recently published with Tamás. The title of the paper is Thornback Mechanism of Small Extracellular Vesicle Release via Limiting Membrane Rupture of Unblock Release Amphisomes, that we refer to as amphiectosomes. And to explain how this whole project started, let me start with the fact that a couple of years ago, we looked at real paraffin-embedded tissue samples from colorectal cancer patients. With my colleague Gábor Waltz, we had a look at these colorectal cancer patient-derived formaldehyde-fixed paraffin-embedded specimens. And what we found was that the cytokeratin-positive green fluorescent tumor cells had some protrusions on their surface. These red fluorescent structures were partially protruding from the surface. They were partially embedded inside the cell. And then with super resolution microscopy, we could figure out that they had some smaller inner structures that corresponded in size to the small EVs. And we looked then at a colorectal cancer cell line, the HT29 cell line in vitro, and we found the very same phenomenon, that from the surface of these cells, these big protrusions emerged. They had inner structures positive for EV, small EV markers. We could demonstrate it quite clearly, and we could find in the conditioned medium of these cells some structures that were relatively large and had some inner vesicles inside of them. We considered that these could correspond to a large ectosome category that we refer to as unblock release MVB-like EV clusters. And we figured that they were released by tumor cells, since we discovered them to be released from colorectal cancer cell lines and cells in patients. But then this was the step when Tamás stepped in, member of my team, and then now I hand over to him because he will present the data.
So after that, the major question was whether these M-block-released multivesicle body-like EV clusters are specific for the colorectal cancer cells, or it is something more universal. So at the first step, we just picked up different cell lines which behaves differently. So we picked up two HEC cell lines, which are immortalized cell line, but not tumorous. We choose two tumorous cell line, the HEPG2 and this same HT29, the beating cardiomyocyte cell line HL1, and the primary bone marrow-derived mast cells, which are growing in cellular suspensions. And surprisingly, the secreted large multivesicle EVs were found with transmission electron microscopy in each case. So all the cell lines secreted similar structures that we found before in case of colorectal cancers. In addition, we found not just these multivesicular EVs, we also found how they bud from the cellular surface. And Most interestingly, we discovered that these large MVLEVs not just present in the extracellular space, but at some stage, their limiting membrane can be fractured, disintegrated, and the small intraluminal vesicles then release into the extracellular space. This mechanism, when these limiting membrane is fractured and the intraluminal membrane is released, we call Thornbag mechanism. The next question was whether these MVLEVs are only present in cell lines or they are available and can be found in different tissues as well. At this stage, we picked up different mouse tissues like mouse kidney, and in mouse kidney ultra-thin sections, we found them principally almost everywhere, both the MVLEVs, both the Thornbag mechanism. We also found smaller EVs inside the tissue as well, and we also found empty large EVs with the released small EVs. The situation was very similar in case of the liver tissue as well. The MVLEVs was present in various type, and interestingly, we were also able to capture inside of a capillary a leukocyte, which is just release these structures. Then we were interested in whether these large multivesicular EVs, MVLEVs, whether are the same structures as we found before in case of the colorectal cancer cells. So we started to test the classical SCV markers, CD63, CD81, ALIX, TSG101, and we found that inside of these large vesicles, both of these markers were present and had a spotty-like structure. For these experiments, we used a PAM-GFP expressing HEC cell line, because with the help of this cell line, we were able to follow the plasma membrane of these large multivesicular EVs, and it's helped to recognize them. We also found the different steps of the Thornbag mechanism with this confocal microscopy study. So we found intact large multivesicular EVs, and we also see how the limiting membrane was fractured and how the small intraluminal vesicles were released. Next, we also tested an autophagy marker, SE3B, and very surprisingly, these MVLEVs contained the SE3B marker with a spotty-like structure in addition with this classical small EV markers as well. However, if we compare the normalized fluorescence spectra of the images, it was a little bit different compared with the small EV markers because the dots didn't overlap. So in this stage, we discovered that the SE3B marker and the classical small EV markers didn't overlap. To quantify these findings, we did a colocalization study where we did colocalization between the classical small EV markers and, of course, these small EV markers colocalized at a high level. But as we also saw from the fluorescent spectra, the autophagy-derived SE3B marker and the classical small EV markers didn't overlap. Therefore, probably they are present in different EVs. The study was continued with additional cell lines. We went back to the HT29 cell lines to check whether the Thornbag mechanism is present there or not. And in agreement with the previous findings, we found the SE3B marker, and we were also able to image the different steps of the Thornbag mechanism. Similar findings was found in case of the HEP-G2 cell line. Both the small EV markers, both the autophagy marker were present. Here I need to mention that these two cell lines, the HT29 and the HEP-G2, didn't express the PAM-GFP protein. Therefore, we need to use a different technique based on lactoferrin labeling to visualize the external membranes. And with the fluorescent labeled lactoferrin on fixed samples, we were able to show only the external membranes without labeling the internal membranes as well. We did colocalization studies with the HEP-G2 and the HT29, and the findings were similar. So the classical small EV markers overlapped and colocalized, while the classical small EV markers didn't colocalize with the autophagy dependent SEC-B marker. The major problem was with the confocal microscopy studies that the resolution of this technique is not enough to detect single EVs. So we went back to transmission electron microscopy. First, inside of the cell and in the extracellular environment, we followed these certain vesicular structures. So we found typical multivesicular bodies, autophagosomes, amphiectosomes, a hybrid vesicle, amphysome in a hybrid vesicle, and all which derived from multivesicular body and an autophagosome fusion. We found in the extracellular space the secreted MVLEV, and the transmission electron microscopy with isolated purified small EV fraction purified from the conditioned medium of the cell. At the same time, we used this similar material for immunogold labeling to test whether these protein markers were they located in these structures. And in agreement with the literature and the previous findings, in case of the multivesicular body, the CD63 was present in the interluminal vesicles of the multivesicular body. In case of autophagosome, the SC3 was in the limiting membrane of the autophagosome, and CD63 was also found inside. In amphysome, we found SC3 positivity and also CD63 positivity. In the secreted multivesicular large EVs, we found interluminal vesicles which were CD63 positive, and we also found SC3 positivity. And very surprisingly, we found in the purified EV fraction both SC3 positivity and both CD63 positivity. Then the question came whether the Thornburg mechanism is an artifact of the sample preparation for transmission electron microscopy, or it's a real phenomenon. Luckily, both the microscope companies Zeiss and Nikon supported us to do live cell super resolution imaging with structured illumination microscopy technique, and with a new cell line where, in addition to the PALM GFP, we were able to express the SC3 protein with an RFP. We were able to follow these vesicles, how the small EVs came out. And within a couple of minutes, the SC3 positive signal came out from these larger vesicles without any fixation, without any artifact, with a live cell imaging. So it's provide evidence that the Thornburg mechanism is existing. So after that, we designed our model to have some understanding how these mechanisms can go through. So based on our ID, the multivesicular body and the autophagosome can be fused and start to form an amphysome inside of the cell. And during the amphysome formation, the internal membrane, the original internal membrane of the autophagosome will be fractured. And during this fracturation, disintegration, the membrane fractures form smaller vesicles inside of the maturation amphysome. So the matured amphysome will contain SC3 positive smaller EVs, smaller vesicles, and CD63 positive vesicles originated from the multivesicular body. Later, this amphisome can be secreted by ectocytosis, and in the extracellular space, it can be present as an exact and intact vesicular structure. As this multivesicular large EV structure is originated from amphisome and released by ectocytosis, we term them amphiectosomes. And later, the limiting membrane or membranes of amphiectosome will be disintegrated, and the intraluminal vesicles released to the extracellular space and will be present in the small EV fractions. With super resolution STED microscopy, we were also able to find the different points and different vesicular structures inside of the cell. So we found multivesicular body where we still used this PAM-GFP expressing hack. So its limiting membrane was GFP positive, most probably through the recycling endosome and this membrane recycling. And inside of these multivesicular bodies, CD63 positive intraluminal vesicles were found. We also detected autophagosomes. These autophagosomes had the LC3B positive membrane, but it was GFP negative. It was also detected when these two structures were fused, and most importantly, we were able to capture the point when the internal autophagosome membrane was disintegrated and the smaller internal intraluminal vesicles were formed. So here we have the maturating amphisome with the GFP positive external membrane. With the cyan color, you can see the rest of the internal membrane originated from the autophagosome, and inside the smaller intraluminal vesicles are present together with the CD63 positive EVs originated from the multivesicular body. And finally, we found the matured amphisome with the GFP positive membrane, and inside CD63 positive smaller EVs and the SC3 positive EVs as well. The question also came where the limiting membrane of these amphiectosomes came from, whether it's really an ectocytosis or there's something different behind. And the question is especially important because with the classical transmission electron microscopic images, these multivesicular large EVs not always show double membrane structure. Luckily, in case of the bone marrow derived mast cells, we were able to capture when this amphiectosome released. It's clearly visible that the cytoplasmic part of this amphisome has a single membrane, but the extracellular part contains both the membrane of the amphisome and the plasma membrane derived membrane. In case of HEX cells, we use the special labeling technique to provide evidence for the plasma membrane origin of the outer membrane of these amphisomes and the amphiectosomes. So what we did, we used a non-fluorescent HEC cell line, and the living cells were labeled with green fluorescence with gamma-glutamine. After labeling, we washed out the excess of this dye, of this fluorescently labeled protein, and then allow the cells to grow additional 3 hours. After that, we fixed them and did an immunocytochemical reaction with SC3B and CD63. What we found that the releasing large multivesicular EVs had a green fluorescence in the limiting membrane, which provide evidence that it has a plasma membrane origin because the plasma membrane was labeled with this fluorescent protein before the fixation. Later, we did additional transmission electron microscopy, especially focused for the question of the numbers of the membranes of these MVLEVs. And in most of the cases, we found evidence for double membrane structure, which refers that at least at a stage of these MVLEVs, the double membranes are present. So after that, we were curious about whether this is a spontaneous release process or it is an active process. So some of the possible control mechanisms were modulated, and we expected that with different modulation experiments, we can follow the release dynamic of these amphiectosome release. So with cytokalism B, we inhibited the actin system. With colchicine, we modulate the microtubules. With chloroquine and bufilomycin, we inhibited the autolysosome formation. And with rapamycin, we accelerated. And then with the confocal microscopy technique, we counted the released MVLEVs and then normalized for 100 cells. What we found, the cytokalism B did not affect the release of these MVLEVs, but colchicine and rapamycin reduced the release dynamic. So the cells release lower amount of MVLEVs, while the opposite effect was found in case of chloroquine and bufilomycin. We also tested what kind of small EVs can be found inside of the conditioned medium. We used immunogold labeling transmission electron microscopy, and we found that inside of this purified EV fraction, we have distinct SE3B or CD63 positive EVs, but we didn't find double positive EVs during this study. The same was repeated on ultra-thin sections, where we were picked up releasing points and released amphiectosomes. And in the intraluminar vesicles, we had the same finding. So the intraluminar vesicles of these amphiectosomes were either CD63 positive or SE3B positive, but double positive intraluminar vesicles in the amphiectosomes were not found. The size of these EVs were also measured based on the transmission electron microscopy images. Between the SE3B positive and negative populations, we found some size differences, which may originate from the different compositions of the membrane. However, it's still a question whether this size difference has any biological meaning or it's just there. In addition, it was a big question how we can detect these SE3B positive small EVs with an independent technique. We did Western blot analysis, and the conditioned medium of the cells were centrifuged with different speed, and we just used the small EV fraction. But after that, after we separated the larger EVs by centrifugation, we purified the total protein of this cell culture supernatant. And then this total protein from this conditioned medium, we did a Western blotting, and we found that in each cases, the SE3 two bands were present. So we probed these blots with SE3B antibody, and the SE3 protein has two forms, has a soluble SE3-1 and has a membrane-binded SE3-2 form. And this membrane-binded form was always present, and as the conditioned medium contains EVs as membrane source, it's provide evidence that the SE3 positive small EVs were present. In addition, we treated with chloroquine these cultures, and we got the same effect that we get during the EV modulation studies. So through the chloroquine treatment, the concentration of the membrane-bounded SE3 increased, and it's provide evidence that more amphiectosomes were released, and from more amphiectosomes, more SE3 positive EVs were present in the conditioned medium. The question always comes whether we find a similar EV structure that we found with transmission electron microscopy, and the migrasomes with their pomegranate structure, with the internal small EVs inside the bigger EVs, looks very similar. Based on the definition of the migrasomes, these are typical for migrating cells, and their release are also actin dependent. And the marker for migrasomes are the tetraspanin 4 proteins. So therefore, we tested our system for tetraspanin 4 protein markers. These are the migrasomes, and you can also see how they released. So during the immunohistochemistry with the confocal microscopy, in case of the tumor-derived HT29 cell line, we found migrosome-like structure with the lactoferrin positive external membrane, and in the limiting membrane, the tetraspanin 4 molecules were present. So based on the definition, these are most probably the migrasomes. In case of the HEC PAM-GFP cell line, we found migrosome-like structure with refractory fibers. However, these vesicles were CD21 positive, so it was positive for small EVs. And at the first look, it looked the tetraspanin 4 looked like inside of these large EVs and not in their limiting membrane. If you take a closer look, in case of the HEC PAM-GFP derived large multivesicular EVs, principally the tetraspanin 4 was present inside and not in the limiting membrane. In case of the HT29, similar phenomenon was detected, but as I mentioned in the previous slide, we also found migrosome structures as well, where the tetraspanin 4 was present in the limiting membrane. To quantify these results, we did the colocalization study, where we calculated the colocalization rate between the limiting membrane fluorescence, whether it's a GFP or a lactoferrin-based fluorescence, and the tetraspanin 4. In case of the HEC cells, the colocalization rates were really low, while in case of the tumor-derived HT29, we had two populations, one where the colocalization rate between the limiting membrane and the tetraspanin 4 was high, and another population where this was low, as in case of the HEC cells. So based on these results, we expect that in case of the tumor-derived cell line, we have two populations, one which is belongs to microsomes, and the other which most probably belongs to amphiectosomes. With transmission electron microscopy, we also tested the different populations. The amphiectosome type of EVs are definitely there, and sometimes we can also find migrosome-like structures with retraction fibers. However, it's still a question whether they are migrosome or not, because with this classical transmission electron microscopy, we didn't have any protein markers, so to validate them, further studies are needed. To sum up the project, so we developed a new small EV secretion pathway, where the mechanism starts with the fusion of multivesicular bodies and autophagosomes, During this fusion, the internal membrane originally come from the autophagosome will be fractured, and then form smaller intraluminal vesicles, and the matured amphysome will contains AC3 positive and CD63 positive intraluminal vesicles. Later, this amphysome can be secreted by ectocytosis. We found the plasma membrane-derived outer membrane around it, and then in the exercise space, we can find the amphiectosomes, and from these amphiectosomes, with the Thornbank mechanism, the intraluminal vesicles will be released. So the Thornbank mechanism seems to be a general mechanism. It can be found everywhere where we searched. It's a new and an active EV secretion pathway, and it's a good and further studies we needed to found the balance between the classical exocytosis and the amphiectosome release and Thornbank mechanism as well. Fundings, we are really lucky because our studies were supported by different authorities, and we received lots of grant to finance our studies. The group who worked on the project, I want to highlight my two PhD students who did quite a lot of work to reach these results, my undergraduate students, and the two microscopy technicians who also worked quite a lot. I would like to thank the help of the whole extracellular EV group within our institute, led by Professor Buzás and the other collaborators. And last but not least, I would like to thank you for your attention, and we are happy to answer questions.
And we do have some questions coming in. So thank you very much, Tamás. This was really fun to see. I think some very striking images that provide a lot of proof for the mechanism that you're proposing. I'd just like to ask if you envision any role for fusion, for membrane fusion in these mechanisms. When I look at those you have a multilamellar vesicle that's emerging from the cell. Is it possible that there's some fusion occurring as well?
Can be possible. We do quite a lot of transmission electron microscopy, and we are still working on how the other mechanism can correlate this process. At the moment, we are working on it, so I don't want to tell any unprovided data, but it's possible, yes.
Very good. All right. Well, let's go to the chat box then. And I just want to remind everyone that I have allowed unmuting now. So, so when I call on you, please be ready to unmute your microphone and show your video too, if you'd like to. And then we will, if you haven't put a question in the chat box, please go ahead and do that too. So let's start with Phil. Phil, nice to see you. I want to apologize for the waiting room problems the other week. Sorry that you couldn't get in, but I know you have a few questions. So please go ahead.
Well, of course, this is wonderful work, and needed continues to teach us details about EVs that we didn't know about. My questions are sort of obvious, is that the tetraspanins, some are in common, but typical small EVs have several tetraspanins, and is that also true of the EVs in these sort of pouches? And my other question is the obvious one of whether there's any functional in vitro or in vivo of these new EVs.
Yeah, well, any of us can answer. Yeah, want to answer, or should I answer? Yeah, so indeed. Hi, Phil. Thanks for the question. Thanks for listening to our presentation. Yes, we have the same sort of tetraspanins that we expect to be on any of the cell-derived extracellular vesicles. And we have not conducted any functional studies yet, so we are not there yet. But I would like to emphasize that these studies were made possible by the fact that the samples were in situ fixed by Tamás. So the reason why probably other researchers have not seen this before was that they start with removing conditioned medium, they pipette it, centrifuge them, and it's kind of a torture to those membranes, which will open up anyway spontaneously. We have evidence that after like 40 seconds, these amphiectosome membranes already start releasing the internal small EVs. Therefore, these studies were probably the first to demonstrate that cells, in fact, in many cases, at least how it was demonstrated by these data, release unblock a whole bag of small EVs rather than doing the exocytosis that has been described before for release of small EVs. But I have to admit that functional studies we haven't done yet.
How could you separate them? The classical small EVs and
don't
the
separate
EVs.
them. So what we do, we have a sticky surface on a cover slip, and we study the cell with their microenvironment. So we don't need to separate them. And that's why we see them very well.
So the fixative is
the larger vesicle has both of them in it, doesn't it?
That's right. But so we are not doing any pipetting, any separation. As Tamás said, we have the cells cultured on these adhesive coated surfaces, and then the fixative is added, and everything is fixed in situ, so without any touching, any manipulation. That's why we see these early events, and that's why we can trace that the cells, in fact, release these big bags in a whole as a total, and then later on, extracellularly, these big bags will be the outer or limiting membrane will be fragmented, and the inner vesicles will be released to the extracellular space.
Yeah, and especially in case of transmission. And especially in case of the transmission electron microscopy, it's very important that we avoid any pipetting during the fixation. So what we do, we have the cells attached to the sticky surface containing glass cover slips. They are growing in the cell culture media, and we add a high concentration of butyraldehyde, which will equilibrate the final concentration without washing, without any additional pipetting. And once we fix the cells overnight in 4 degrees, then we start the sample preparation. And principally, with this way, the structure of these large multivesicular EVs can be preserved, and they stick to the environment where they are, and you can keep them.
The last question is, do these exosomes express any unique surface determinants compared to the small EVs, sometimes known as exosomes? For separate.
At the moment we don't know, but we are working on it. So we try to purify them later, and then we are planning this kind of marker studies.
Well, by your history in this wonderful group, I think you're going to get there. Thank you.
I hope so.
Thanks for the questions. Next question is from Esther.
Hi.
Yes.
Congratulations on the nice work, and it's nice to see fellow Hungarians being active in this field. My question is, I think you partially answered it already, probably, but I was interested, how long do these vesicles survive after they detach from the cell? And I guess a follow-up question is, if you can imagine this entering the circulation and transport the exosomes, the small vesicles, to like distant sites.
Yeah, so as I pointed out when Tamás presented, actually by live cell microscopy, you could see that as early as after 40 seconds, the limiting membrane of these large multivesicular amphiectosomes start to open up, and the inner vesicles, the exosomes, start to escape to the extracellular space. This whole process is, for sure, is completed by 5 minutes. And the other large EVs are reported to have much longer lifetime. So, for example, for microsomes, and by the way, for microsomes also it has been described that one possible fate of this microsome that they have a fragmentation in their plasma membrane and the inner vesicles are released. But for the microsomes, the lifetime is reported to be 400 minutes.
Yeah, but in the other hand, it's also an interesting thing that with the tissue transmission electron microscopy, we clearly see that they are in the circulation. However, we don't know where they come from sometimes. So if you remember, when it's just released from the leukocyte, it's clear where it's come. But when we see these MVLEVs inside of the circulation, we don't know where and when they come. And what we also see sometimes, that independently whether the limiting membrane of the large EV is fractured and some of the small vesicles came in, sometimes there are some residual in the broken envelope. And in that case, it may transport it. It's something that we also want to study in the future.
Exciting. Thank you.
Thank you. Next question is from Ming Lin.
Okay, can you hear me? Yeah.
Yes, thank you.
Okay, good. Congratulations for your wonderful work, showing a lot of very wonderful images. You know, the dynamic, is a fusion or release of these membrane vesicles need power, which come from the cytoskeleton. Of course, you shows some a slide shows using inhibitors. Have we get a chance to look at the using the cells with fluorescent labeled cytoskeleton, either live cells or not? That will show more beautiful images showing the dynamic changes.
Thank you for this question, and I think it's a very good suggestion. At the moment, we didn't do that, but in the future, I think it's something that we can try. Yeah, so this is a relative new study, and we're still working on it. So one of my PhD students hopefully will defeat her thesis from this topic as well. So yes, I think we should try this. It's very useful.
Okay, good.
Thank you.
Thank you. Next, we have Clotilde.
Hi. Thanks. Well, yeah, very nice work. I was wondering if it's possible to evaluate whether when you isolate the EVs from the fluid, I mean, doing all this very careful way of not breaking the bags before and so on, whether you can evaluate how much of the EVs come from this torn bag mechanism versus EVs could come from amphysome fusing with the plasma membrane, which was the original way we thought of amphysome-derived vesicles. It's very difficult to do. I'm not sure how I would do it, but do you have any idea?
Thanks, Clotilde. If I can answer this question, this is very important. I think we are not contradicting the previous studies because fortunately we already have quite promising data showing that in certain circumstances exocytosis does occur. So these two mechanisms in parallel exist. So we have this torn back mechanism and then also we have quite substantial evidence for the conventional classical exocytosis, exosome release pathway from cells. And we hope to present these data at the ISAB meeting. So in the biogenesis session, hopefully you will hear about these novel findings. And you asked about the proportion of the LC3 and CD63 positive EVs. Well, already we have some again promising data in collaboration with Tijana Talisman, and so depending on the sample source, of course, this may vary, but I can tell that the LC3B positive vesicles are also quite significant.
Thank you.
Thanks, Clotilde. Naveen.
Thank you. It's an excellent talk. So I'm like a new postdoc. I'm pretty much new to this EV research. So my question is, I just initially missed few of the slides. So my question is, this amphisomes and amphictosomes, is it a separate mechanism of EV release, or whatever EV is released to make this mechanism has any separate clinical relevance, which are better, like EVs are better, or is EVs released to amphictosomes are better? So what is your thoughts? Thank you.
Yeah, thank you for the question. And yes, this is a very important issue and a very important question, and we are working on it. So what we are doing now, we try to compare human samples under different pathological conditions and some controls. And we want to follow whether we found any clinical relevance or not, or what can be. So we are working on this. Of course, I cannot tell any data about it because we just started, but we hope that there will be some clinical relevance, but it's ongoing.
Thank you. Thank you very much.
All right. Any last questions? This is your chance. And if not, I hope that we see many of you in Vienna in just 3 weeks. The ISEV 2025 Meeting Education Day will be underway at this time in 3 weeks. We expect to have over 1,700 people in attendance. I hope that you will be among them. If you haven't signed up yet, there's still a chance. You can still register.
Excuse me, I have one question.
Yes, go ahead.
Not related to the talk. I'm from US. I'm an immigrant, so I cannot travel everywhere. And past 3 years, this meeting was happening everywhere other than USA. So when it is coming back to USA?
So we were in Seattle 2 years ago.
Yeah. After that, it was never in USA. So I learned a lot. Actually, I started my EV research by coming to that meeting. So after that, I wanted to attend this meeting. Because of the visa things, I could not travel. So I'm interested to attend this. So I want to know when it's going to happen again in US.
So we have a geographic chapter rotation for the ISEV annual meeting, and it goes from the Americas chapter to the European chapter to the Asia Pacific chapter. Not necessarily in that order, but we do a rotation. And so we will be back in the Americas. I don't think that we've disclosed yet whether it will be in the US or another country in the Americas, but that will be next year.
Okay, thank you.
In that respect, will there be a Zoom aspect of the Austrian meeting, so that a lot of it might be covered?
For the overall meeting, there is not. Unfortunately, it ends up being something like planning 2 meetings to have both a Zoom meeting and an in-person. However, there will be specific sessions, especially with some of the task forces, that will have Zoom components. So please stay tuned for the information that will be coming out about those. So we do want to offer opportunities for people to participate in some of the events remotely. So thanks for the question.
Thank you.
Yes, and the Education Day talks will be recorded and will be made available online on the ISEV's YouTube channel.
Thank you for everybody's answers. Thank you.
All right. Well, thank you so much, everyone. And I see there's one more comment here from Pascal, who says, "Very nice work. Indeed, would be interesting to have some quantitative info on the different pathway contributions." So I'm sure we'll be hearing some more about that coming up. All right. Well, thank you everybody for joining today. Thanks especially to Edit and Tamás for the excellent presentations, and hope to see you all again very soon. Take care now.